Morning everyone. Good to see quite a few of you online already. It's really good. So we're going to carry on with Biology 7 today. I keep wanting to call it Biology 3, but Biology 7 today, so we're going to keep going on about ecology. So we've got online, good morning Nico, Ruby, Tanishka, Lily. Anyone else online? Say hello. So we've looked at, so far, we've been looking at the ecosystems, we've been looking at a lot of the terminology that comes into ecosystems. We've looked at food webs, we've looked at food chains, and we're going to sort of go on and look a little bit later on at biodiversity and bring all that back together again by looking at the sort of human impact and the human effect on that as well. So we'll come back to that when we get there. Morning Amy, morning Jack, morning Grace. What I want to do to start off with today is we need to look at how we can actually measure populations, how we can study different communities. Now, I know if you've looked at any of the questions that I posted in the classroom last week, some of the questions were specifically around using quadrats, around transects, and looking at ways of sampling and doing population counts. So I sort of set those up for you to have a look at, and we're going to go through today, and we'll sort of look at some of those answers, and we'll see how you could have answered them as we were going through. Now, it's really important, excuse me, I'll just sort of look over this side for a minute, I'll just load up something else. It's really important that when we're actually looking at sampling populations, that whatever we sample, we do in a random way. I've seen it on a field before where we've had students that have gone out and they've just mowed this field. And we were going out originally to do sort of a daisy population count. So the students have gone out with these quadrats, and a quadrat is literally a ½ metre square. You'll have seen them in school with a grid in the middle. And they allow us to estimate population counts. But they only allow us to estimate a population count if we use them in the correct way. Unfortunately, this particular day, like I said, they'd mowed the fields. Students went out. Where are we going? There we go, that one there. Students went out with their quadrats, and they went and found the only patch of the field that hadn't been actually mowed. And they sort of threw this quadrat down over the daisies, and there were sort of 4 daisies in the middle of this quadrat. So they then went through and they did something else that we do, and they multiplied it by the area of the field. And they came up with this sort of idea. I think there was something in the region of 10,000 daisies on this field. And you're sort of looking around the field and it's completely empty because they've all been mowed, apart from these sort of 3 daisies. And it was because actually they wanted to desperately count some daisies, but because there weren't any, they chose the only patch to throw the quadrat down in. And their calculations indicated, like I said, 10,000 daisies, which would have been really nice. It would have been the field completely blanketed with daisies, but there wasn't any other daisies in sight. So what I want to do is I want to have a look at ways that we can actually measure and estimate, and all of this is estimation counts. Remember, the only way that we could actually do a proper population survey and get the exact number would be to individually count each individual organism. And in most cases, unless we're looking at things like trees or maybe big animals, you would expect a farmer to know the exact number of sheep or cows in their field. But if we're looking at most things, we can only come up with an estimate. We can't come up with a true value. But we can come up with a fairly accurate estimate. So let's make a start with this one. So the first thing we're going to have a look at is using quadrats. Now, I said before, there are these quadrats, and they're not just ½ metre squares. I think if you looked at the questions I gave you last time round, and I'll show you a picture of that in a moment, we've generally got some form of grid. Just put that down there. And they're sort of separated up into squares. I'm not going to do this. I really apologise to those of you that like everything nice and neat. I probably should have planned this beforehand. The squares aren't going to be the same size, so a few of you will have to just cope with that for the moment. But we get the idea that we've got this grid filled with all of these squares. Now, the first thing that we would do with this one is we would throw it randomly. And I'll show you another, well, I'll talk about another way in a moment that we can, if we don't want to throw it, that's possibly better, that we can look at random sampling. And those of you in a minute, we'll sort of talk about transect lines. So we're going to throw randomly. So obviously in a safe way, making sure there's nobody behind you, the easiest way is just to throw it randomly behind you where you can't actually see where you're going to throw it. The next thing that we will then do is we go through and we count species. Now this could be individuals, or it could actually be the different types. Under the last geography field trip I went on, we went up to Snowdon. Actually, in a lot of cases, what we were doing is we were looking at the number of different types of species within the quadrat, and if we could, the number of each of those as well. And all of this would be then recorded. Now it might... Oh, recorded. It might actually be that there are so many of those species, imagine like grass, that we couldn't count the whole number in the whole of the grid. But what we could do is we could just count the number maybe in one square, and then we could multiply it by the grid so we could work out the number in that particular area. We're then going to repeat. Now, realistically, you would repeat as many times as you physically could, as you had time for, in different areas, different parts. So if it was a field or a woodland, you would do it in as many different varied areas as possible, still throwing it randomly, because then we can work out an average. So we would work out our mean, and then we could multiply by the area of the field. And in most cases, that works really well. And I think what they're after... Now, that was a Transect line. Do we have one with Quadrats? Yeah. Let me just show you this screen, if it's going to work. This was one of the questions that I set you last week to have a look at. And if you notice, they placed a 1 by 1 metre Quadrats. This is bigger than the one I was talking at before. At 10 random positions. And they counted the number of dandelions in each of the Quadrats. They then went through and said, why do they place it in random positions? So it's to avoid bias. And by bias, I mean, if you think about those students I was talking about at the start, when they were doing the daisy calculations, they were really biased because they desperately wanted to count some daisies and there weren't any on the field apart from those 3. So it avoids bias. And it's only a 1-mark question, so I wouldn't even write a sentence. I would just put avoids bias. And if you look at the mark scheme for that, there you go, avoid bias. So that is all you would need to write down. We don't need to write big paragraphs and sentences for that one to get the marks. In order to estimate the total number of dandelions, remember it's a 5-mark question, and we can see that down here. Now, that means there's going to be multiple calculations that's involved in this one. The easiest thing is to break it down into small steps. So what I would be doing at this stage is I would go through and I would calculate the mean. So I would add these up, and I would divide by 10 because there's 10 samples. And I think off the top of my head, and I haven't done this straight away, this would give you an answer of 60. I think it's 60. Hang on, let me just have a quick look. Nope, 6. I knew there was a 6 in it somewhere. So add them up, divide by 10. The next thing you need to do is work out the area of the field. Now, again, this is one of those things a lot of students start to panic at a little bit, but we can break this down into 2 rectangles. So we can either do a rectangle that is 150 by 200, and then we've got another one then that would be 250 by 100, and we could work out the area of both those rectangles. Remember, that's just Key Stage 3 maths. I think it might even be Key Stage 2 working out the rectangle. The big part is actually working out here which rectangle or square you're going to do. The other way you could do it is to do 150 by 300 and then 100 by 100 square. And then you would add those areas together to get the total area of this field. If you then multiply that by the average number of dandelions, you would get an estimate of the number of dandelions on the whole field. And if we have a look at the mark scheme here, we should get a value of 330,000. And do they ask for standard form? Yeah, give your answer in standard form. So, in order marks, if you've done this one, you can mark your own. We would get a mark for the correct mean. We would get a mark for the correct area. You get a mark for multiplying the mean by the area to give you, and that's not including the answer. You then get a mark if you get the correct answer on this one. So remember, this one here is just a mark for doing the calculation correctly, writing down the calculation. So when I say about you've got to write down your working out, this is why. We get a mark for getting 330,000, and then we get a mark for converting it into standard form. So those are where the 5 marks come from. So see how you did, and then afterwards we'll do the Google chat as normal, and you can tell me how well you've done if you come online with that one. The next one then is then to plan an investigation. So they're looking at light intensity affects the number of dandelion plants that grow in the area. So how would you investigate that hypothesis? Now, for me, the area is this area of woodland. If you think about the fact those trees are going to create some shaded areas that's going to restrict the light coming in. So we're going to get some low light intensity areas here. And we're going to get some higher light intensity areas here. So the easiest way of doing it is you're going to need a light meter, or I would use a light meter. And all I would do is in the randomly placed quadrats, you could also measure the light intensity in the areas that you place the quadrat. Now, if we want to be really good with this one, we can do the next bit, which is to use a transect line. Now, I'm just going to go back to my whiteboard for a minute, and we'll come back to this in a moment. And we can look at actually how we could use a transect line. So let's have a quick look. Oh, there we go. So a transect line is literally a tape measure. So let's just imagine, technical drawing now. There's my tree. Okay, so we're going to get some low lights here. And we're going to go out to higher light levels here. And what we're going to do is we're going to run a tape measure out from the tree. So let's just call this our tape measure. Now, you would normally decide what distance you wanted to do. And some of this comes down to the size of the quadrat. So if our quadrat is 0.5 metre by 0.5 metre, we want to place this at set intervals. Now, although we're choosing where we're placing it, because the tape measure is dictating where those quadrats go, this still counts as a random sampling method. So I'm going to place it every 2 metres. So I'd end up placing one there, one there, one there, and we'd work our way down, all the way down to the end. This would be a 2 metre gap, this would be a 2 metre gap, and so on all the way down. We could then count the number of dandelions in that area, and what we are going to do is we're also going to record the light levels. Now, you may want to record other things as well, because actually we could record the other a lot of other factors, and we can actually then measure and sort of come up with some other hypothesis or some other conclusions. So we may want to, let's just think about, we've obviously said lights. Well, we're obviously going to count the dandelion population in that area. Dandy. Where was my exam question again? Dandy. There we go. But we also may want to measure moisture of the soil. We may want to measure pH probe. So we literally just shove the pH probe and the moisture level into the soil and see what these are. And we record all of these. We may not necessarily use them to straight away. We may only want to look at the light, but it means we could come back and look at those sort of samples afterwards. And as scientists, actually, I'd be inclined to measure all of these, put them in a table and see what sort of patterns we can see. If we're looking at sort of coniferous areas, so we're looking at fir and pine, I know they actually change the pH of the soil, which makes it harder for other tree species to grow in there. So we may want to look at this as well. We could take soil samples, and we could then sort of look at minerals, or we could look at sort of biomatter, and I'll come back to biomatter when I do the carbon cycle a little bit later on, sort of biological material. We could do wind samples, so we could do all of these sorts of things as we're working our way out. But the key thing is we wanted to look at light, and we wanted to look at the dandelion population, and then all of this would be recorded into a table. So when we're looking at using transect lines, it's quite a nice question to get. I know it looks quite complicated at the moment, but there's only about 4 or 5 bullet points, and you just adapt those bullet points to the question they have set you. And you would just literally write them in another way. You'll see when we do evolution, we've got the same thing. There's just a set number of bullet points, and it's the same bullet points for every single question. So we're going to lay out the tape measure. We're going to place the quadrat on the tape measure. For this one, if I'm doing light, I'm going to record the dandelion population, and I'm going to record the light level. And I may want to number these. 1, 2, 3. Move quadrat 2 meters along tape measure. Repeat 3. Okay, and then actually what we would just do, continue until end of tape. That does say tape measure, honest. Okay, so we're going to lay it along, we're going to move along. Morning, Raoul, good to see you online. So we're going to continue along. Sometimes you can actually sort of group some of these a little bit into sort of more than one, or into sort of one bullet point. Generally, though, the one thing that a lot of students miss that they quite like putting down afterwards is to move the transect and repeat. Now, this could be move it to a different tree, it could be move it further along the field, if it's a seashore it could be move it along the beach. But they really like in the exams to move the transect to a different area and repeat so that we can get a mean results, because this avoids bias and it helps us identify any anomalies that we might have in our results. So those are the sort of steps that you want to put in when we're doing it, when you're doing a question based on this. And if we go back and have a quick look at the question on this one, we've got placing the quadrats, we've got the random, some way of achieving a random way. You go along a transect line, regular intervals, that's our 2 meters, different areas of light intensity or the transect running along, so we've got that one down. For each quadrat, we're going to count the number of dandelions, we're going to measure the light intensity, and then we're going to compare the data. That's pretty much most of the steps that we had down there. If I go back up to, and we'll look at a different question, let's have a look at the seaweed. So this one, they actually tell you what they were doing. So we've got a 50 meter tape measure running down. The quadrat was placed next to the tape measure, and we recorded whether species were present or not. The students placed the quadrat at regular intervals to achieve random. Again, it's to avoid bias. And it's to make sure that actually we have a random test. How could we improve it? Well, we could repeat it at different points along the beach. In terms of the graphs then, what they're looking at, these sorts of graphs come up quite a bit. Now there's some really... Okay, so Tanisha, the quadrat and the transect, they have to get used together. So the transect is just another word for a long tape measure. And then the quadrat is a grid that you put next to it and you move it along. But they literally get used together. But there's no point in having a transect line generally unless you've got the quadrat to go with it. There is a way of doing it where you lay the transect and you just see if there's any species touching the transect. But generally we use a quadrat to go alongside of it. Okay? Right. Let's come off of that for the moment. But in terms of sampling, these are the two methods. But it generally comes down to trying to achieve a random sample to avoid bias, okay? And those are the sorts of things you're trying to get with this. Right, let me bring my notes back up again. Any questions for that one, pop it in. Otherwise, you can ask me afterwards when we go through and I do the Google Meet. But it is important that you can look at random sampling. There was another question I was looking at actually with this, and I was trying to find it to see whether we can actually, so I can go through. It's quite difficult when you want to try and work out a population study of animals or insects. And the reason is because you've got those animals and insects moving into and out of the area you're moving. And sometimes they can move quite big distances. You need a way of trying to work out whether you've already counted an individual species before. Things I've seen come up in exam questions is they were trying to do, say, a population sample of beetles in one particular area. So what they would do is they would actually have a small trap, which would be like a pit in the ground they can't get out of. They may sort of set it up one day, test it the next day, count the number of beetles, and then maybe mark each one with, say, a little drop of nail varnish or a colour that isn't going to affect them at all. They would then do the study the next day, and they would then count the ones that don't have that particular marking on them, and you could repeat that over several days, and you could then get an idea of the population in that particular area, and you could do the same sort of study in different parts and different places. But like I said, when you start getting to some animals that may move big distances, working out the population gets quite tough. There's been a study recently where, so stag beetles are quite endangered, and there's actually a website that you can go onto at the moment. We've actually found one fairly recently, but I'll show you a picture of it. We found one in our garden. They're trying to encourage people to always sort of leave, if you've got a garden or an outdoor area, try to leave sort of small piles of dead wood to try and encourage the stag beetles because they are endangered. So what this website has done is, rather than trying to find ways of capture them and do population studies that way, what they've done, and here you go, I'll just put the picture up so you can see, this is the one we found in our garden. What they've done is they've actually got the public now to go through and basically record whenever they see one, where they record, upload pictures, so they can get a population study and they can try and find out where they are, how widespread they are. So this is a male stag beetle. Interestingly enough, they can't actually hurt you with these big pincers. They only really use it for attracting the females and for when the male's fighting, because they're actually so big that they can't get enough strength into them to actually cause any damage to us. Whereas the females ones have much smaller ones that can actually give you a bit of a nip. But the males ones can't. But yeah, this is one we actually found in our garden. Let's have a quick look. We'll just come off that for a minute. I've got a picture of the female as well. No, this was another one of the male on a log. Hang on, let me put this back up again. And they're quite cool little things. Lots of people don't tend to like bugs, but they just literally get on with it. We literally, in our garden, I dug a little tiny nature pond that's, it's literally about, well, it's tiny, a couple of feet by maybe about sort of 30 centimetres deep, and I just put a piece of plastic liner and then planted some plants around it, and it's quite interesting what you actually get in there. Let me just see if I've got a picture of the female. I might not. We did see one, just so I can show you the difference. There we go. So this is the female stag beetle. And you can see actually its pins at the top are very, very tiny. But yeah, this is, in terms of sampling, again, this is just a way they're trying at the moment to get people to basically do a population study for them. So the only reason I'm sort of saying this is you never quite know what they're going to ask in an exam question. Yes, you have to be able to do these bullet points I spoke about over here in terms of using a quadrat. I have seen Brave Wilderness, yeah, it's quite cool. And in terms of using a transect. But it's also worth thinking outside the box as well. It's actually worth thinking a lot more about what other questions could they ask you. How could you do studies on maybe sort of more rare species? If you think about some of the birds of prey, some of those can be in quite remote places and we don't have that many of them. So how could we actually try and improve? There's another sampling question that came up talking about different birds into the gardens. And the problem is then that the study might very well depend on the time of the day, the time of the year, because actually this time of year we've got quite a lot of birds out there, but as we go through, you may find you get lower populations if the birds are nesting, for instance, to try and, they're sitting on eggs. It may be a case of actually you see more of them in the evening when they're trying to catch insects. It might be a case of actually it's too early in the year and the birds haven't actually migrated in yet. So when you're looking at studies and we're looking at doing sampling, it's worth considering different factors that could affect the population that need to be considered by the scientists or by the conservationists that are actually doing those population studies. Okay, so yeah, I've just been going on a little bit about that, but it's just to try and give you an overview that it's not sometimes just as easy as throwing out a tape measure and going through and counting those population studies. Okay. Right, the next thing I want to look at is the carbon cycle. Absolutely, Lee, yeah. Absolutely type of food. It would depend on the environment. So, for instance, if you were to look at maybe a garden that backed onto woodland, you may actually be able to count the number of deer that you see. If you had it onto fields, you would have a different area. It might be that you've actually only got a patio in your garden with pots. So therefore, the type of insects that you get would depend very much on what would live on those particular pots in those particular plants. If you have trees overhead, you may then be able to do a population count on squirrels, for instance, which you wouldn't if there were no trees in your area. So all of these sort of factors, as scientists, we need to consider. And sometimes this comes down to the research that you do before you do the study. So what you may want to do if you were looking at a particular species is you would be spending time on the internet, you're spending time in libraries, looking in textbooks, trying to find out what type of habitat, what type of ecosystem. Remember those keywords biotic and abiotic that I mentioned in the last couple of videos? What type of biotic and abiotic factors affect the organism? Because once you know all of this, you can then start looking at the areas they might be in and then start planning your study accordingly. So when we were looking at the seaweed study, for instance, some of those seaweed will have the air bladders in them to float to the surface because they actually need to be able to get close to sunlight. When you look at some of the other sea creatures, they actually cope very well with being battered. I mean, you look at limpets on rocks. It's quite easy to do a study of limpets because actually you can lay the transect out and you can go and count them on rocks because that's where they feed. Because actually when the tide's out, they just sit there in one place and they can actually sort of glue themselves and suck themselves onto the rock. But when the sea's over them, they'll move around and they will use the sort of bottom to eat off the algae and the various minerals and food on those rocks. But because they can actually attach themselves so firmly to the rocks, they can actually live quite comfortably in areas where they're getting battered constantly by the waves. Whereas you look at some of the other more delicate creatures and some of the snails, some of the oysters and some of the other sort of creatures, they maybe can't cope with that level of battering without getting detached. So you probably wouldn't find them in those areas. But if you've done your study beforehand and you've actually looked at the conditions they need, the food types they need, and the conditions they need for their habitat and their ecosystem, you can then sort of tailor it, hopefully still trying to make it random. Like I said, if you were to go and look at an area, if you're looking at limpets or another type of sea creature and you knew where they like to be and you only measure that area, it may not give you an accurate idea or an accurate population count for the whole of the beach. For instance, you wouldn't find limpets generally on sand because they can't actually get their food on it and they can't actually attach themselves to sand properly. They need rocks. So if you were only looking at the sort of the rocking area and then you said actually you had this beach, let me just move over. If I just go down here, if we had a sort of beach area where we maybe had some sort of sand and then sort of some rocks and some rock pools here, if we just sort of go through maybe some rock pools and then the sort of sea went out and then this area here was sand. I don't think I've got a colour for rocks. Let's just do them brown for a minute. This may be our rocks. Then actually, if you only looked at this area down here for the limpets, then it wouldn't give you an accurate population of what's up here. So we actually need to take an account as well. And if we ran our transect, let's just go back. If I ran a transect out, say, this way, and let's just call this my transect line, something else that I may... Oh, hang on, done that again, press the button. Something else that I may want to record is actually the terrain. So I may want to record in my table that this is sand, this is rock, this is a rock pool, got another rock here, this is another rock pool, And I know those of you that do geography, what you'll actually be asked to do is sketch pretty much a diagram of sort of a sideways view. I can't remember the technical term they call it. And that labelling all these things on. And it sometimes only takes a matter of minutes, but you're going to label all this on, and you can do this on the diagram, and then you may want to do your population counts as you're working your way down it. Yeah, spot on. So Lily, so the little air bladders in them help them float to the surface, so they get lots more sunlight, so they can photosynthesize quicker. So actually, and I know you've probably chosen your options, there's a massive overlap with the geography and with science in these particular areas. And it's sort of an area that if you know one subject, you can use this and you can apply it to the different subjects as well. We went out, I think it was last Wednesday, bizarrely enough. So unfortunately, my kids always wake up early. So we decided that we were going to plan to go out to Cowshot Beach. So I live sort of near Eastleigh. We were going to sort of plan to go to Cowshot Beach early in the morning. So we got to Cowshot Beach at 6 AM in the morning. As you do. We've got a little sit on top kayak, so we took that out with us as well. We dragged the kids out there. And the tide was completely out, and I've never seen it out quite as far as it was. Which means in most places, until you got about 100 metres out from the shoreline, the water was probably only about 20 centimetres deep with a bit of pockets. There was a sort of groin going out as well that you could walk along. So we had areas of seaweed, we had areas of sand, we had the rocky areas, and we had the areas of this sort of big groin that was going out. We did eventually sort of get far enough out to have a bit of a paddle and a bit of a swim. But actually what we spent most of the time doing was looking along the groin. We looked in the sort of the seaweed areas and the sandy areas. And we were looking at different types of crabs. We saw hermit crabs, we saw spider crabs, we saw some little fish, we saw some mud skippers. And we saw all of these different species that you wouldn't normally see. Like I said, 6 AM in the morning. And we were literally going along with my kids, like I said, they're 6 and 8. And we were just looking at the different and sort of talking about why they would live in this area. Why might we see more crabs in maybe the seaweed areas? And it's the sort of thing that you could do really, really easily. And then, yeah, we still managed to get my son back to school for 9:20 when he started school. So, yeah, it was quite cool. But these are the sorts of studies that you can do. And you don't actually need any equipment for that one. You can just look at, actually, why would this particular organism be living here? And those are the sorts of questions the exam board's going to be asking you to do. Thinking outside the box. As Lily's just said, with the seaweed and the air bladders. Why might some have air bladders? Well, it helps them float. They're going to be on the surface, got a bigger surface area. They're going to get more sunlight, which means they're going to be able to photosynthesize faster, which means they're going to grow faster because they're going to be able to produce their own food at a faster rate. So for them, they're going to be able to outcompete the other species. Brilliant. Right, let's move on and we'll look at the next part of this lesson. So let's have a look at the carbon cycle. So the carbon cycle is all to do with getting carbon out of the atmosphere and putting the carbon back in. Now, the diagrams we produce on this, there's lots of different diagrams. If you look online, and I'll show you what I mean quickly. Let's just go on here. If you literally just go through and type in GCSE carbon cycle and go to images, there is loads and loads of different carbon cycles on here that you can have a look at. Some of these will link you through to other websites. Some of these will be better than others. So I'm going to show you how I would do this one. But if you're looking at it afterwards and go that a bit confused, or you want to have a look at what other people have done, don't be afraid to go and have a look at what other people have actually produced. Because I've seen 2 or 3 versions of the carbon cycle come up on exam papers, and every single one of them is different. So it's not even like there's a standardised form that the exam board will use. Okay, so just go through and have a look and just get familiar with looking at different styles. Now, we're going to start off with carbon dioxide in the atmosphere. And there's a few overlaps here with different subjects. There's going to be sort of combustion coming in from chemistry. There's also going to be the look of sort of dissolving in the oceans and chemistry of the atmosphere as well, which is, again, another chemistry topic that we'll look at doing later on in the year. So there is definitely some overlaps between the different sciences. And the better you are at looking at those overlaps, the easier you'll find some of these subjects. So the first, and probably nowadays, the most important method is that photosynthesis. Okay, so we're just going to put that down there. There's the arrow. So this is going to be carbon being taken out of the atmosphere. And if you remember when we looked at this earlier, this is to do with plants taking out the carbon dioxide. They break the bonds because it's a chemical reaction, and they then use the sun's energy to break those bonds, and they make new ones. So they turn that carbon dioxide into glucose, into cellulose, into proteins, into fats, into all the different substances the plants need to grow. Because remember, when we eat those plants, we break down those dead animals and plants, and we use those molecules to build our bodies out of. But to start with, plants have to synthesize those molecules. The other thing that can happen to the carbon dioxide is it can dissolve into the oceans. Now, this still happens today, and there is a bit of an effect with the sort of temperature out there as well. So as the temperature changes, we can affect the amount of carbon dioxide that dissolves into the oceans. This was much, much bigger when the atmosphere, when the Earth sort of, in the early days of the Earth, when the atmosphere first formed, there was huge amounts of carbon dioxide. And as it cooled down and the oceans formed, lots of carbon dioxide dissolved into the oceans. And that then went on to form rocks such as limestone. So limestone itself formed sort of a massive carbon sink. And we can actually have over here, I'm going to just do a very small arrow because actually this isn't as big nowadays. But it did go on to form limestone. And actually, when we go and produce concrete and cement, if I just put down concrete and cement here, that is part here of the carbon cycle. And we also have, during that production method, more of it is actually released back into the atmosphere. This is a bit that not many people put into the carbon cycle, but it is actually a way that we get carbon back into the atmosphere. So the next thing we've got then is from the plants. There's two things that can happen. Three things technically. Oh, there we go. They can be eaten. They can be burnt. That's a U there. It's actually quite hard writing on a tablet because you can't move around as much. Or they could die. Okay, so this is death. The other thing that will happen as well, if I can see if I can do this. Right, that's a rubbish one. Hang on. The other thing that can happen, let's see if I can just move it here. No, I really can't draw these arrows today. That'll do for a minute. Is we've got respiration happening. Okay, because remember, respiration doesn't just happen in animals and bacteria, it does happen in plants as well. So the animals can respire as well, and the animals can also die. So we've got combustion of plants, we've got the respiration of animals and plants, we've then got this sort of forming detritus, so we've then got our decomposers and our detritivores. In other words, worms, insects and species that eat detritus. Now these will also put carbon back into the atmosphere by respiration. So we've got all of this respiration happening as well, sort of pumping back into the atmosphere. Now if you notice, if we just take this one section here, this is carbon neutral. In other words, there's no... Okay, sorry, let me just move that over for you. Is that better? If you notice, there's no general increase in carbon dioxide going either in the atmosphere or out of the atmosphere. We take it out by photosynthesis, and then all of that carbon dioxide that's taken out goes back in either by combustion or by respiration. So there's no overall increase. This is why actually burning wood on wood burning fires is better for the environment than burning fossil fuels. because the plants take in the carbon dioxide as they're growing, we release it when we burn it, and then it gets taken back in by more plants. The problem we've got over this side is we can also have our fossil fuels. So, creation of concrete and the burning of fossil fuels increases the carbon dioxide. So these are not, if I just do a line down here for a minute, this is the carbon neutral section. These are not carbon neutral. So this adds extra CO2. Okay, so this adds extra CO2. So this is good. This is bad because this is increasing the carbon dioxide, which is then adding to the greenhouse effect. Now, what a lot of what some companies are starting to do now is they're actually planting additional trees each year in certain parts of the world. So as those trees grow, it takes in the carbon dioxide, and they're trying to offset their carbon production. So I've had an email recently. I do a bit of web design as well. So the company that I use for my hosting, they've actually started paying for trees to be planted, I think in Madagascar, paying for trees to be planted because they know that the rate of those trees that grows will take in extra carbon dioxide. So the cost of running their servers, so the electricity and the carbon dioxide production, is offset by these trees that are being planted. So actually they can turn around and say at the end of the day they are a carbon neutral company. And this is where we need to try and be if we're going to reduce the effect of of global warming, or stop it getting any worse. Now, I'm going to come back onto this a lot more in the next session when we talk on Friday. Okay, so I'm not going to go into that now, but it's important that you can make sure that you're happy with how carbon dioxide is taken out of the atmosphere and all the different ways that it's put back on, and also how it moves through a food chain. So this bit, by being eaten by animals, we could then have eaten by other animals, and so on. But this is enough that we for us to have a look at here. If I go back onto this bit again, we can start seeing, if we just sort of pick a few of these at random, they've pretty much got, they haven't necessarily put respiration, or they put respiration on a few times, but there's different ways of showing the same thing. See, here's one I've missed up here, volcanoes. So we can put that on as well. The burning of fossil fuels, by the way, but I'm going to put volcanoes on because I quite like having that one. Let's put that one over here. Just gone behind my head again, but we can have that one on there as well as the volcanoes erupt. Remember that the burning of fossil fuels in combustion, that's not necessarily just in power station, that's in cars as well. And pretty much anything that involves either methane, oil, coal. Okay, I think, looking at this, that is most of what I need to cover for that today. Next time round, what we're going to have a look at is we're going to have a look at decomposition. We're going to have a look at decomposition. We're going to have a look at the effect of humans and also natural effects in terms of biodiversity. And then we're going to sort of go through and we're going to link that through. There's another required practical that you need to know, and that's looking at how we can measure the rate of decay of milk. Specifically milk, although we can use the same ideas as we sort of move our way through. So, as always, in fact, I'm going to do it now while I'm talking to you guys. I'm going to be on Google Meet pretty much as soon as this call has finished. So I'm just going to create a meet called Science. So literally, no excuses with this one. If I just click on Join Now, the link code is going to be on Google Classroom. Now, Year 10's gone on. Year 9's gone on. Okay, so hopefully I'll see you on Google Meet in a few minutes. Take care. If not, I'll see you Friday.